So hello friends, welcome back to my channel Medicine 101. Today we'll talk about glycolysis part 3. Okay, so before starting the video, let me tell you guys, I have already uploaded the two videos based on glycolysis in terms of glycolysis part 1 and glycolysis part 2. Okay, so it's my request to you, first watch the two videos, then come to this video. Otherwise you maybe will not be able to understand the whole topics over here. Okay, now before starting the topic, let's take hypothetical cells where we will see the glycolysis process, and I just briefly mention about whatever the thing we discussed in the last two videos. I mean in terms of the glycolysis part 1 and part 2. Okay. Okay, now see. For example, is the cells over here. Okay. Now I have already said you how the glucose will come by the facilitated transport and then the active secondary active transport within the cells. And we know there are different types of in terms of the facilitated transport by the blood transporters through which we will get the glucose within the cell. Now whenever we get the glucose within the cells, for example, I'm just taking the glucose over here. Now I will represent the glucose in terms of the 6 carbons. So for example 1, 2, 3, 4, 5 and 6. Now see, in the first steps we already talked about whatever the thing we'll see, it's the unidirectional pathway. That means it's the irreversible. Okay, here we'll see what the glucose that will be converted into glucose 6 phosphate. Okay, let me write it over here. So 3, 4, 5, 6, and for example it's a phosphate molecule over here. Okay, so it will be what, glucose 6 phosphate, and two types of the enzymes we did talk about here, that only hexokinase that is present in almost all cells. And what about the glucokinase? Now I already did the comparison in between the hexokinase and glucokinase in the part 1 video. Yeah, okay, so in the part 2 video you will get it, these things, the comparisons in between the hexokinase and glucokinase in part 2 video. Okay, now see the glucokinase that is present basically within the liver and within the pancreatic beta cell. Pancreatic beta cell over here. Okay, after that whatever the step we'll get then the reversible steps over, or reversible, we'll get the reversible steps where we'll see the glucose 6 phosphate that will get converted into what, fructose 6 phosphate. Fructose 6 phosphate. And we know there's a glucose and fructose. If I just change in terms of the structures, they are what, isomer of each other, so you can write it over what type of the enzyme that will act over here. That is what, phosphoglucose-isomerase. It's not that much important step. Phosphoglucose-isomerase. Okay, now you have to know that the product, we know that, just try to understand from here, from the first step we will get from the glucose to the glucose 6 phosphate. Now this will be the product. Now this glucose 6 phosphate that can inhibit the enzyme what, hexokinase. It can also inhibit over here. And what about glucokinase? The product we are getting over here, what, the fructose 6 phosphate. Okay, this can inhibit the glucokinase. So try to understand the glucose 6 phosphate that can inhibit the enzyme hexokinase, and fructose 6 phosphate that can inhibit the glucokinase over here in the okay. Now the most important step of the glycolysis process, the most crucial or important, whatever you can say, the unidirectional pathway. Let me write it over here clearly. Okay, now we'll get what, from the fructose 6 phosphate we'll get fructose 1,6 bisphosphate. Get fructose 1,6 bisphosphate. Okay, now see, we're getting fructose 1,6 bisphosphate. Now what type of the enzyme we'll get over here? The name of the enzyme is very important, I'm saying, so listen to it very carefully in this step. Okay, now name of the enzyme is phosphofructose-kinase. Phosphofructose-kinase 1. I will write it over here on. Why I am writing on, I will let you know. Okay, within a couple of minutes we will come to know automatically. Now whatever thing specialty we will get in these enzymes. Now the phosphofructose-kinase 1. Okay, now basically when it's enzymes, so enzymes, they are what, they are derived from the proteins. Proteins means whenever we see the structures of the protein, the formation of the proteins, at that time we will get in tertiary structures, we will get the different types of the foods within the proteins. So we can consider the foods over here in terms of the pockets. We'll get the different types of the pockets in terms of the what? Phosphofructokinase on over here, okay? So 1 pocket, 2 pocket, 3 pocket, for example, just like this, okay? Now the first pocket is the site ATP binding site. So the ATP can bind it over here. Then the citrate that can bind it over here. And AMP, adenosine monophosphate, that can bind it over here. Now why I'm saying so these things, just try to understand that by just whenever these three things will get bind with the phosphofructokinase on, at the time the phosphofructokinase on that can understand whether it is supposed to do the glycolysis process forward or not. Okay, see, whenever within the cells, try to understand, whenever within the cells the energy is less, at the time we need energy. So whenever we need energy or requirement of the energy in the cells, at the time the glycolysis process should go forward. Okay, now see, whenever the ATP that will be less, and see if the whenever the energy is less, definitely the citrate will be less also, because citrate is the part of what is the substrate within the TCA cycle. So if the TCA cycle continues less, as a result we will see less amount of the energy, so definitely the citrate that will be also less over here. And try to understand from AMP. Now adenosine monophosphate, whenever the ATP will get breakdown into adenosine monophosphate, that means the concentration for the AMP that will be more, because whatever the ATP was there, all of the ATP that got broken down into what? AMP. Now see, this ATP and the citrate, they will act as what? Whenever the concentration is less, okay, whenever the concentration is less over here, now the whatever thing we'll see over here, the cells are supposed to do the glycolysis process, so they will act over here as a negative regulators, okay. So whenever the amount is less, as a result, they are supposed to say the glycolysis process should be started over here. So the negative regulators. And what about the AMP? See, whenever the AMP is more, that means the ATP is less. That means the AMP that will act as what? The AMP that will act as, the positive regulators. That will act as the positive or regulator. Okay. Now these are the things over here. Okay. Now I'll talk about one more important thing over here. For these things I have to enlarge space over here. So let's enlarge space. Now see, within the enzymes we are getting the three types of the substrates, or we can say the things they are getting attached with the phosphofructokinase on. Now definitely there should be some phosphofructokinase or something like this. Now whatever the thing we'll see. Now if I say, for example, is the phosphofructokinase on, for example is the phosphofructokinase on, just like this one. Now I have already said you guys that there is binding site for ATP, there is binding site for citrate, there is binding site for AMP. Okay, so citrate is the binding site for what? AMP. Now the most powerful stimulus, I'm repeating once again, the most powerful stimulus to activate the phosphofructokinase 1, that is what? Fructose 2,6 bisphosphate. Now I'll say what is this thing also. Okay, now see, the most powerful one, that is fructose 2,6 bisphosphate. Fructose 2,6 bisphosphate. Let me write it clearly, then you can understand these things part clearly more. Okay, for this thing use this part over here. Okay. Now for example, okay, in this place. Now is the phosphoglycerate on. Okay, the enzyme organized like this. Now for example, is the site for the ATP binding, is the site for citrate binding, is the site for AMP binding. Now the most powerful signals or the most powerful stimulus will get over here, that is what? That is for fructose 2,6 bisphosphate. That means fructose 2,6 bisphosphate is the most powerful signal for these things. Now see, this is having the site for phosphofructokinase over here, and whenever it will stimulate this on, the glycolysis process would go move the glycolysis process that will go forward. Okay, sorry for this. Now whenever we will get the fructose to 6 bisphosphate, that will come over here and that will stimulate the fructose phosphofructokinase 1. As a result we will see the glycolysis process that will move forward. Okay, now how the fructose to 6 bisphosphate is coming? See, from the fructose 6 phosphate we got the fructose on 6 bisphosphate. In the same way, from fructose 6 phosphate we will get also what, the fructose to 6 bisphosphate. Now for this thing we will get the enzymes. Now the enzymes that will locate over here in terms of the bifunctional enzyme over here. Okay, bifunctional enzyme, that means the enzyme that will convert the fructose 6 phosphate into fructose 6 bisphosphate. That is what, that is the bifunctional enzymes. That means we will get over here the enzyme that is fructose, okay, let me write it over here, phosphofructokinase 2, phosphofructokinase 2, that is converting the what, fructose 6 phosphate to fructose to 6 bisphosphate. Here is 2, that's why we will get fructose to 6 bisphosphate. Here is 1, that's why we will get what, fructose on 6 bisphosphate over here. Okay, now another side we will get over here, that is known as phosphatase. This region that is known as of the enzyme phosphatase. Now how the enzymes are regulated, that's the important thing over here to know. Now for this thing I have to modify a little bit steps for these things. For that okay, I'll show over here. Okay, see, whenever I am talking in terms of this enzyme, that means try to understand, I hope you understood already from here that the phosphofructokinase 2 that will convert the fructose 6 phosphate into fructose 2 6 bisphosphate. Okay, and along with that we will get the, as it is the bifunctional enzymes, we will get along with that the phosphatase part of the enzymes, another part is. Okay, now see, for example it's the bifunctional enzyme, it's the bifunctional enzyme, for example is the phosphofructokinase 2 part over here, and is the phosphatase part. Phosphatase part over here we are getting. Okay, now see, during, if I say whenever we need energy, that means whenever the glucose is less, that means glucose is gone, that means whatever the hormones that will be activated, that is the glucagon hormone, right? So see, for examples from the GIT, the glucose, whenever we will see the glucose is less, as a result we will see the glucagon hormone that will be activated, and for the glucagon hormones we will see there is certain types of the receptors over here for the glucagon where the glucagon can bind. So whenever the glucagon will bind over here, okay, glucagon is the receptor, is the G stimulatory receptors, and this can activate the adenylyl cyclase, and whenever the adenylyl cyclase that will be activated, that will convert the ATP into cyclic AMP. Okay, so actually let me repeat once again, the whenever the glucose is less within the, whenever the within the circulation or within the cells the glucose is less, as a result the glucagon hormone that will be activated, and whenever it will be activated, it is having the certain types of the receptors, G stimulatory receptors. Okay, it will come and it will activate the G stimulatory receptors. Whenever the G stimulatory receptor will be activated, it will activate what, the adenylyl cyclase. The adenylyl cyclase that will do what, it will just convert the ATP into cyclic AMP, and whenever we are getting the cyclic AMP, that can activate, okay, protein kinase A, and whenever we will see the protein kinase A that will be activated over here, that will come and that will do what, the phosphorylation, that will do the phosphorylation in these regions, that will do the phosphorylation of these enzymes over here. Okay, so this is the process we will see in these cases. And whatever the insulin, whenever the glucose is more within the cell, whenever the glucose is more within the cells, the ATP is good enough amount of the ATP, there is no problem in terms of the ATP, in terms of the citrate, in terms, okay, but whatever the thing we'll see in these cases, we'll see, for example, whenever within the cell, try to understand from here, within the cell, if we see the energy that is good, energy is enough, that means the ATP that will be more, the citrate that will be also more. But what about AMP? The AMP that will be less. So whenever the AMP by the positive regulations, it will stimulate the what phosphofructokinase 1, and phosphofructokinase 1 that will be what inhibited over here. That means the glycolysis process that will be not started. In the same way, the ATP and what by the negative regulations, whenever the ATP and the citrate that will be more, by the negative regulations, what they will just inhibit the phosphofructokinase 1 enzyme. As a result, we will see there will be also inhibition of these processes. So there will be no what glycolysis further in terms of the forward reactions over here. Okay. Now see, I am repeating once again that phosphofructokinase 1, this is the enzyme, it is having the certain types of the binding sites. For example, it is having the binding site for ATP, it is having the binding site for citrate, it is having the binding site for AMP, and the most important one, that is it is having the binding site for fructose 2,6 bisphosphate. And the fructose 2,6 bisphosphate that will come from where? The fructose 2,6 bisphosphate that will come from the fructose 6 phosphate. And there is the presence of one type of the enzyme, bifunctional enzyme, that is known as phosphofructokinase 2. The phosphofructokinase 2 that will convert the fructose 6 phosphate into fructose 2,6 bisphosphate. And whenever you are getting these conversions, that will stimulate the phosphofructokinase 1. We will see further what all type of the reactions over here. That means in terms of the forward reactions of the or glycolysis. And whenever during the feeding conditions, we will see the reverse things. As a result, we will see over here, for example, is the receptors for insulin. Whenever the glucose is more, insulin will give the signals. As a result, we will see what they will remove these reactions over here. That means they will remove the phosphate groups from here and here also. Okay. Now the rest of the things in terms of the glycolysis we can see over here. So this was all about the third part of the glycolysis. Then the subsequent part will meet you. Okay. That means in terms of after that what type of the reactions we will get in terms of like we got the fructose 1,6 bisphosphate, then we will get the dissociations of the two ways. I mean in terms of the phosphoglyceraldehyde and dihydroxyacetone and phosphate, then what type of the reactions we are getting over in details we'll talk about in subsequent parts. Thanks for watching. See you in the next video. Don't forget to subscribe.